That was good. Sharing, recording. Control Alt. I got rid of that damn thing. Hit escape, it comes back. That's pretty good. Looks okay? Yeah. Okay, let's see. I think it's 11 o'clock. Morning, everybody. It's what, Thursday? Thursday, March 18th, 2021. And you guys are biology 1160 section 2, Tuesday, Thursday class. And what I remember from last time, we stopped right before we got to the extrinsic eye muscles, right? I mean, we were talking about the eye. These are accessory structures of the eye. And, you know, there's different ones. We talked about eyebrows and eyelashes and so on and so forth. Extrinsic, I think the conjunctiva, did we do that one? Well, anyway, we're up to number 5, the extrinsic eye muscles. Here, I tell you that there's a total of 6. Let me get this right here. Total of 6 skeletal muscles per eye. There's 2 oblique muscles and 4 rectus muscles, all for voluntary movement of the eyeball. And we talked about these when we were talking about the cranial nerves. remember there's 3 cranial nerves associated with voluntary movement of the eye. There's cranial nerve number 3 was the oculomotor nerve, and it innervated 4 of the 6, and you can see which ones are right here. But it also includes the levator palpebrae superioris muscle, which is not really moving the eyeball, it moves your eyelid. But in addition to that, the superior rectus, medial rectus, inferior rectus, and the inferior oblique are all innervated by the oculomotor nerve. And I'm trying to do something here. It's not, there it goes. This would disappear. Okay. And then when I told you about the trochlear nerve, cranial nerve number 3, you know, you get 12 cranial nerves, and they all like do all kinds of special great things, but the trochlear nerve only does one thing. It innervates just one of the 6 muscles, and that's the superior oblique muscle. And it's named after this, see that little loop right there? That's called the trochlea. It's like a ligament that supports this muscle, kind of allows it to kind of come in at a strange angle right there. But anyway, that's the trochlear nerve innervating the superior oblique muscle. And then the 6th one, which was innervated by cranial nerve number 6, the abducens nerve, that innervates the lateral rectus on one side. So what you're looking at right here, this is someone's right eye. So their nose would be situated like right here. And so when this muscle contracts, it pulls the eye in that direction. And you can see it, look, right. Oh, yeah. Also, where is it labeled? This is looking down, the superior view. Somebody's right eye, and there's the lateral rectus right there. And so, you know, the center of the face would be here, the side of the head is over here. And so that makes sense. This muscle right here, when it pulls, it's going to pull in this direction, and it's going to cause the eyeball to, you know, go like that. So you abduct your eye. Okay. So there you go. And they're all considered accessory structures of the eye. You know, they move the eyeballs, but it really doesn't have anything to do with seeing per se, like detecting light or anything, just directing the eyeballs. So anyway, extrinsic eye muscles. So there you go. That's all the accessory structures. Now, remember we talked about the anatomy of the eye as a whole. You have accessory eye structures, and then the eyeball itself, or it's also known as the ocular bulb, the visual apparatus of the body, what detects light. And I know eyeball doesn't sound very technical or anything, so but that's what we call it, or the ocular bulb if you want to be more anatomical. All right. So what's the eyeball or the ocular bulb made out of? And like you probably would guess, there's always 3. Remember the number 3? There's always 3 layers, 3 tunics, 3 whatevers. And of course, there's 3 tunics of the eye. And we talk about them from the outside in, from the superficial layer, working our way deep. And so the first tunic is known as the fibrous tunic. Think of it be fibrous, right? I define it as the avascular superficial covering of the eye. And I can just kind of stop right there because the fibrous tunic itself is divided into 2 parts, or there's 2 structures that make up the fibrous tunic. First, the sclera. This makes up the bulk of the fibrous tunic, because you think fibrous, heavy duty. That's the outer white of the eye, the sclera. You see that? It's continuous with the dura mater, that structure that covers the brain and spinal cord, that superficial mosa. Remember the 3 meninges? And remember I told you if you remove the skull cap, you won't see the brain immediately. You actually see this white kind of membrane. That's the dura mater. And that's the same thing. If you want to see what it looks like, you know, look at the white of your eye. That's dura mater. So it's the same tissue, But here, surrounding the outside of the eye, it serves primarily for those 6 extrinsic eye muscles. It gives them something hard and fibrous to grab a hold of, and pull. You're moving the eyeball, and so you got muscles pulling on it. So it's hard and durable. So that's the white of your eye. That makes up, like I said, most of the fibrous tunic, but it's thick and fibrous, and consequently it's opaque. The light can't pass through it, so you got to modify it somewhat, and so it is modified. The fibrous tunic, the anterior-most portion that covers over the iris, is clear. All right, and so that clear portion is called the cornea. The cornea, defined as the clear anterior extension of the sclera, suspended from the surface of the iris by an anterior chamber, which is filled with aqueous humor. So what I'm telling you is that here's your cornea, and then there's a space in there. And see the space they're pointing out? The anterior chamber. Now there's just not air in there. There's fluid, okay? And you have fluid throughout your body. And depending on what it's doing and where it's located, it'll have different names. Like just body fluid in general is often just referred to as interstitial fluid. The word interstitial means literally between the tissues. But this fluid, it's derived a lot the same way cerebral spinal fluid is derived from like a choroid plexus. You have these leaky capillaries located here and here that produce it. So really it's cerebral spinal fluid, but it's not in surrounding the brain and spinal cord and stuff like that. It's in this anterior chamber, but it's the only place you find it in the eye. So we call it aqueous humor instead of CSF, but there it is. It's located right there. And it's continuously, a lot like cerebrospinal fluid, it's produced from the blood and it's reabsorbed back into the blood. So it's kept fresh all the time. Okay, so there you go. So that's the first tunic, the fibrous tunic. Now the second tunic, remember tunics or cloaks are like layers. Remember, I think the analogy I gave you is like the layers of clothing a Catholic priest would wear. They wear like these capes. I guess you call them a cape. And underneath that, there's another layer, and then another layer. And each thing means something. Well, likewise here, the next layer, deep, means something. It's something completely different. Functionally, it's completely different. So we call it the vascular tunic. The vascular tunic is more commonly referred to as the uvea. The word uvea means grape because it kind of looks like a dark black round grape, okay? I define it as the middle tunic of the eyeball, and it in turn is divided into 3 functional regions, and we'll talk about those. The first, the bulk of the uvea, is called the choroid. The choroid accounts for all except the anterior 15% of the uvea. It is highly vascular and highly pigmented. It forms the dark chamber, this dark posterior chamber of the eye, basically like a photo chamber. And you can kind of see this middle layer. I guess it's kind of pink colored in this illustration. But in life, it would be very, very, very dark, almost black. Like if you dissected an eye, I don't think any of you have ever dissected like a cow's eye in like a lab or something, that's really dark pigmented area. And that's where all the blood vessels are located, in the choroid, okay? But the idea with all the pigments, the dark pigments, it makes that back part of the eye like a photo chamber. I don't know if you guys remember back in the day when cameras used to take film. Remember that? You had to buy film for a camera. Well, you open up the back of the camera and you got to load the film, et cetera. Well, the back of the camera where the film is going to be exposed to light when the shutter opens, called the photo chamber. And it's always, always, always flat black, okay? You take an art class, learn about your colors. Black is the absence of all color. White is all colors combined, believe it or not. It's like when you're seeing colors, what you're seeing are wavelengths of light. You see blue, you're not seeing all the other colors are being absorbed except blue. You see purple, you know, all the colors are being absorbed in this thing I'm holding, my mask, except purple, and that's what's being bounced out at you. When you see white, all the wavelengths are being bounced back at you. But when you see black, all the wavelengths of light are being absorbed by my phone. If you're looking at what you're looking at, okay? And so if you want something to detect light, you want it to be able to absorb light. So it's black in there, okay? Anyway, that's your choroid. Now, remember that's the 85% of the uvea or whatever, everything except the anterior 15%. So what's the anterior 15%? Well, the ciliary body. If you move forward, you kind of get into this kind of a muscular area here. See this pink material? Part of it's the ciliary body. Ciliary body is an apparatus to modify the shape of the lens. If you look right here, of course, you can see the lens, just like a lens in a camera, a lens in a microscope or magnifying glass or whatever. It magnifies and it changes the angle of light, and you can focus on things. That's why you have a lens, so you can focus on whatever, right? So you can see. It has smooth muscles associated with it, which are called ciliary muscles, and it's suspended. It's acted upon by suspensory ligaments, which in turn are attached around the perimeter of the lens. I'm about to sneeze. I keep playing with my nose. I don't know, I guess it's because I'm looking at light. Is that what it is? You look at light, you want to sneeze, or you look at light not to sneeze? I always forget. Here it comes. Excuse me. All right. I need a drink. Okay. Well, what's going on with this? And here's I included a photo, a scanning electron micrograph of a lens, and you can see this is the lens. Don't they come in pairs? Sneezes. Excuse me. There you go, right there. You see these little fibers around. Those are the suspensory ligaments, and the suspensory ligaments are attached to the ciliary muscle here and here. And so, what is this doing? Well, to focus, there's two ways you can focus by using looking through a lens. One, have you ever played with a magnifying glass, like you fried ants on the sidewalk or whatever? Well, you know, I don't want to encourage you to be cruel to nature, but anyway, I think everybody's played with a magnifying glass. And you know, if you want to look at something, so how do you get it into the focus? Well, you move the lens either closer to or further away from your face, right? That's how you get it into focus. That's one way you can focus with the lens, by changing the distance from which the lens is to your eye. Okay, that's one way. Another way you can focus a lens is change the shape of the lens. Like when you get a new prescription. Look, I'm wearing two glasses today. This and this, like built-in cheaters. Anyway, you get new glasses. You get a new prescription in your glasses. What does that mean? Well, the lenses are a different shape because they're focusing different because your eyes changed. So here, to focus on something, the lens changes shape. Like if you want to look at something far away, the ciliary body contracts and it pulls on the suspensory ligaments, and the lens kind of stretches out. So you look at something far away, it stretches out. And when you want to look at something up close, it relaxes and it returns to its thicker shape, and so you can see something up close. And it works beautifully. It stretches out, goes back, stretches out, goes back, whatever. But it only has about a 30-year warranty. What? What does that mean? Well, for about 25, 30 years of stretching out and coming back, stretching out and going back, it gets kind of like overstretched, and so it doesn't come back all the way. And so you know what happens when you're about 30-ish years old? You gotta wear these. You gotta wear cheaters. Yeah, because if you don't wear cheaters, what do you have to do in order to see something? You can't change the shape of your lens anyway because it's stuck in a kind of like a far-sighted position. So what you do as you move it away, remember you change the distance. You can read the newspaper like that, or read the instructions on your, how do you cook macaroni and cheese again? How many cups of milk or whatever, or just buy you some cool cheaters like I got today. Anyway, but that's all this. Wasn't that fun? Now we're talking about the vascular tunic, right? There's more to that anterior 15%, and that includes the iris. Now the iris, the word iris, it's also the name of a flower. You think flowers are colorful and pretty, and so I guess this is the colorful, pretty part of your eye. That's the colored part of your eye. It's also known as the iris. And but what is it? It's not there to be colorful and pretty. That's not its function. It's here I tell you that it is the colored portion of your eye. It's continuous with the ciliary body. If you look carefully, here's the ciliary body. That's muscle, and then you kind of see some more muscle extending forward as the iris. Okay, it contains smooth muscle to regulate light in the eye by controlling the diameter of the pupil. Remember the pupil, the dark spot right there in the center? It's not a spot, it's actually a hole. It's a hole that's in the iris that allows light to pass in and enter that photo chamber, just like I think you call it an iris on your camera, right? Isn't that what you call it? The is that what you call it? The shutter, that's what you call it. This is like a shutter. It's closed. Well, not on you, it's never closed. There's always a little bit of opening, but the larger it gets, the opening, like the smooth muscle, it allows it to dilate. It allows more light in. That's what of course happens in dim light situations. When you don't have a lot of light to see, it expands to allow as much light in as you can to see in the dark, okay? But if you're outside on a bright sunny day like today, you're going outside, it constricts and gets very, very, very small because you don't need a lot of light to come in. There's too much light. It's like, overstimulating. So you get a little pinhole pupils, and just plenty enough light to enter the eye and stimulate and form a image, right? So there you go. That's all your iris right there. So that's our second layer, the vascular tunic. Now, the deepest layer, it's called the sensory tunic, or some books will call it the nervous tunic, because that's the sensory portions. Remember, that's what we're talking about, special senses, right? But honestly, nobody calls it the sensory tunic or the nervous tunic other than an anatomy professor. Most people are familiar with this layer of the eye, and they simply refer to it as the retina. And what's the retina? Well, the retina, that's the functional portion of the eye, the actual visual sensory organ, okay? And it contains the cells that actually detect light, and they're collectively referred to as photoreceptor cells. But there's two types of photoreceptor cells in the human eye, and the names are associated with their morphology because they have distinct shapes. You have rods and cones. Of course, the rods are kind of rod shape, and the cones kind of have a cone-shaped cell body, but they're functionally, they're different. The rods, these are by far the most numerous of the photoreceptor cells. These are the ones that are functional in dim light and are responsible for peripheral vision. Like if you're looking straight ahead, you're looking straight ahead, you can kind of still see things on the side, if not even like turning your eyes that direction, right? That's peripheral vision. All right. Your cones, these are the ones that are active in bright light and are responsible for color vision. To see in color, the cones are associated with that. And those are situated and more concentrated right behind the pupil. That makes sense. If you're looking straight ahead, here's your pupil. You get a beam of light coming through straight like this, and bam, it strikes right here in the back of the eye. So you got most of your cones right here, and then the further away you move from this bullseye, we'll call it for right now, the fewer cones you have. And so up in this area here and this area over here, this is peripheral vision. So peripheral vision here, peripheral vision here. And if up in those areas, you would have just mostly rods. But your cones are like right there, all right? Now, structures associated with the retina include that bullseye I was telling you about. It's called the macula lutea, which literally means the yellow spot. I guess I've never seen it look yellow. If you're looking inside somebody's eye, maybe, I don't know, at some point it looked yellow histologically or something. I'm not sure why they call it the yellow spot. That's that bullseye right in the back, right? It's right behind the pupil. Okay, it's an area of high concentration of photoreceptor cells. The number, like I said, the number of cones increases proportionally from its periphery towards the center. You get more and more cones, the closer you get to the center, until you're right smack dab in the center, which is called the fovea centralis. That's the area of the macula lutea where you have just pure cones. Okay, so the purest, just all color vision, the most detailed vision, whatever, is right there at the fovea centralis. Okay, so that's what's going on right here. And then another structure, noteworthy structure in the back of the photo chamber, the eye there, is the optic disc. You see this right here. You see where all the blood vessels are radiating out from one location right there. You see that? Now, if you look into the posterior chamber of somebody's eye, it's not going to be all colored like this. It might be yellow and have red vessels and blue vessels. It'll all look kind of like, it looks like to me, it's kind of like lightning. It looks like white, kind of like branching lightning or whatever coming out. Anyway, but if you trace any one of these vessels back, they all come back to the same point right here. And that's the optic disc. The optic disc is also known as the blind spot because there are no photoreceptor cells in this area where it's attaching. On the back side of the retina here, that's where the optic nerve is attaching, and running down the center of the optic nerve or the arteries and veins traveling to supply blood to the uvea. So what you're looking at, you're looking actually through the retina and into the choroid. So that's technically where the blood vessels are. They're in the choroid, and the retina is really thin. It's on one centimeter thick, and it's just like on top of the choroid. Okay, so this spot right here, there are no photoreceptor cells. They're called a blind spot. And when I used to teach the phys lab, we used to demonstrate this. And there's an exercise in your textbook. You can do this just for funsies, I guess. Remember I was telling you about peripheral vision. If you're looking at something straight on, say like you have like a cross that you're looking at like on a piece of paper, and I think they have this in your textbook wherever they're talking about the eye. If you're looking straight on at a cross, you would like that image, that cross would be like right here, right over the macula lutea, because that's where you're looking at. And you cover up one eye and you look at it, you can see it. And then off to the side, if you have like a dot, you can see a dot in your peripheral vision. And we used to do that. And so you look at a cross on a piece of paper and you got like a dot over here on the side, and you see it and you're holding it at a certain distance from your face. And then you start to move it. You don't take your eyes off that cross. You're looking at that cross right here. You can still see that dot, let's say like it's a red dot off into your peripheral vision. And as you're moving with one eye covered, that dot will move. You can't tell that it's moving, but the image, the angle of it is still on your retina. So it's moving. You're still stimulating, all right? But there's gonna be a point where it crosses over the blind spot. And when it does that, in your peripheral vision, it disappears and then it reappears. You come like this, disappears, reappears, disappears, reappears, disappears, reappears. And we used to do that in lab. Oh, wow, look at that. It's novel, fun things we used to do in the lab. Anyway, so that's your eye. So what's next? Our third special sense, taste, or gustation as it also is called. Remember, taste or gustation is one of those chemosensory things. It's a lot like smell. Remember, you're detecting chemicals, and the sensory structures in the cells are pretty much the same, except these in turn are in your oral cavity opposed to your nasal cavity. Okay, so here I tell you the taste receptor cells for taste are chemoreceptors located in the special sense organ, the taste buds, or they're also called gustatory caliculi, or singular would be a gustatory caliculus. These three structures you see here, they kind of look like three onions, don't they? Three Vidalia onions or whatever. But those are the gustatory caliculi, or taste buds, embedded in this case on the tongue. But they could be throughout your oral cavity. We're going to talk mostly about the ones on the tongue. Okay. The histology is a lot like the olfactory epithelium. So the olfactory epithelium is, lining the entire nasal mucosa up in that superior nasal concha. But here you got little pockets of them. They're a little bit different. So you got clusters of cells forming the caliculi. And so I tell you about the cytology. It's the cytology that the taste buds includes basal cells for replacement, which is pretty much the same. You have supporting cells, and then the actual gustatory or taste cells that are actually stimulated, that actually stimulates a nerve fiber that generates an impulse and sends it to your central nervous system where you interpret taste. Okay, so there you go. Now, where do you find taste buds? Well, taste buds are located throughout the oral mucosa, although they are concentrated on lingual papillae, or sometimes it's called tongue papillae. Remember, a papilla in anatomy is anything that's kind of raised structure. I guess it's kind of raised like a nipple, so a nipple-like structure. But there's 4 different shapes or morphologies associated with the tongue papillae. And like I said, most of the taste buds that you have are located, embedded on these papillae, okay? Most of the time. In humans, I think in mammals, there's I think 5 that we usually recognize, but in humans there's only 4. We don't have a particularly special tongue. Tongues vary in mammals and in all vertebrates really, but the tongue reaches its pinnacle in evolution and complexity in mammals, of course, because remember, mammals are the masticators. Those are the ones that chew. And so if you're chewing food, you're manipulating food in your oral cavity. You're just not going like that. You got the tongue in there to help move stuff around. And since it's in there touching the food, it has these chemosensory structures, gustatory caliculi, to give you information about what you're eating. And there's a correlation with the complexity of those caliculi and the animal's diet. For example, the most diverse and complex taste buds are in your herbivores, things that eat just plants. And the least complex and the fewest in number are at the other end of the spectrum of the carnivores. Now you think, why would that be? We think about the carnivore diet versus the herbivore's diet. Carnivores, they eat meat. That's it. They just eat meat. Herbivores, they eat plants. I mean, there's all kinds of plants. And so your ability to be able to detect one type of plant versus another is pretty important. I mean, meat, you can't really screw that up, right? Well, it either tastes like fish or chicken, right? Everything. Well, what's alligator taste like? Well, I like chicken, a little bit like fish, mostly like chicken. How does chicken taste like? Well, ducks or sparrows, I don't know, just meat, I don't know, it's not that complicated. Anyway, humans, we're omnivorous. We eat plants and animals. And so ours is kind of not real special. It's kind of middle of the road. Anyway, let's talk about the 4 classes of papillae. Like I said, I have them listed down here in this little diagram, that's based on their shape. Then the first one, number 1, this is the most common one that's found everywhere, all over the surface of the tongue. They're called filiform papillae. The word filiform literally means filament-like or filamentous, and it's something that's filamentous is skinny or thread-like. Get it? Filiform, like a filament thread. These are the most numerous over the surface of the tongue, and they're added to the tongue for texture to facilitate food manipulation. So that's why they're there, to kind of grip the food, give a nice kind of like a sandpapery kind of substrate to grip and manipulate the food. But consequently, there are no taste buds on the filiform papillae, and that's just true for all mammals. There's no taste buds there. They're there. And of course, from one critter to the next, the filiform papillae will change. Anybody can tell you that. There's a difference between being licked by a dog and licked by a cat. Have you ever been licked by a cat? It's like really rough tongue, because they got really hard keratinized filiform papillae. Anyway, so what about the ones with taste buds? Well, all the others have taste buds, beginning with number 2, fungiform, like a fungus, like a mushroom. So fungiform, something that is fungiform is mushroom shaped, kind of shaped like a mushroom. And I guess this is kind of like a longitudinal section through a mushroom. It's kind of rounded at the top and kind of narrows, like a stalk-like shape at the bottom, I suppose. But these are, here I'll tell you, the secondly most abundant papillae. And they have a moderate number of taste buds. These are the ones that form the little visible little dots you can see over the surface of your tongue. If you stick out your tongue and look in the mirror, you'll see these bright red dots. Here they show on my illustration as little white dots. Maybe some people, they're white. I think that when I've looked, it's like little red dots. But anyway, those are the fungiform papillae. They're not, I guess they're the most visible ones that you could see, kind of scattered all over. And they have, those caliculi or taste buds embedded in the mucosa. Okay. Number 3 are the foliate. Foliate, it means leaf-like, like foliage is like leaves on a tree. So something that's foliate, it's kind of leaf shaped. Does that look like a leaf to you? I guess that looks like a leaf. Leaf shape, foliate. Anyway, these are special papillae located on the lateral margins of the tongue, like on the sides. And these are most sensitive in children. It includes numerous sensitive taste buds on the lateral surface, and like I said, they're most abundant and most functional in little kids. And that's why actually children have a more acute sense of taste compared to humans. I mean, adults, human adults anyway. That's why like kids seem like they're pickier when it comes to eating. They're picky eaters, and then later, you grow up and you mature and you seem you're more sophisticated. You have more of an acquired taste. And, oh yeah, when I was a kid, I couldn't stand this or that or whatever. Remember the first time you ever tasted beer or coffee when you were little? You probably didn't like it, or a martini. You ever tasted a martini? I still don't like martinis, but I remember my mom drinking a martini one time at a dinner party, and it was a cool looking glass, with things sticking out of it, and she's sitting there drinking it. I said, I wanted to experience that. Oh, let me taste the martini. Oh, you're not gonna like it. I just, begged her, and of course, my mom, spoiled me. She's okay, just a small sip. So she hands me this cool, martini glass with the stuff sticking out. Oh my God, I thought I was drinking turpentine. It was terrible. I said, Mom, how can you drink this? It's terrible. It's an acquired taste. When you get older, but
what it is, it's not an acquired taste when you're an adult and you're mature. It's that your taste buds just aren't as good as they were when you're little. I mean, when you're little, you can taste stuff. When you get older, you can't really taste anything. So you eat anything, you drink anything. It just tastes bad. And then same thing when the first time I tried escargot in a fancy restaurant with my my parents again. And that was my stepdad this time, and he's a big fan of escargot, and he went, "Oh, you guys want an appetizer? Let's get appetizers. I'm gonna have escargot." Ooh, wow, escargot! I'm gonna try that. You know, he's eating it. I'm gonna eat it. Brought me the little tray of snails. You know, I dug it out with a little special little fork and put it in my mouth. So, oh my God, I spit it out. I said, "I think mine are bad." He goes, "What? No, mine are delicious." And he was just eating his way. I said, "No, mine are definitely bad." And tasting you, no, that's how they're supposed to taste. I said, "Oh my God." If you would have blindfolded me, I swear you were feeding me out of a full diaper. That's what it tasted like to me. Anyway, enough of that. Our fourth and final lingual papilla or tongue papilla are called circumvallate, or some books just call them velate. Circum just means like circular, and vallate means like valley. They have these deep trenches that go down. These are the large ones, way in the back of the tongue. In most mammals, it forms this like a V-shaped arrangement in the back of the tongue, or maybe a lambda, if you want to call it lambda. But it's kind of, it kind of makes an apex at the center of the tongue back there. You stick your tongue way out, and you look. It kind of almost looks like little warts in the back of your tongue. But these, of all the papillae, these are the least numerous, confined to the back of the tongue. And we used to think, so why do you have, this kind of papilla and that kind of papilla? And there was someone that actually did their PhD dissertation on this. So, well, you have different papillae, they detect different forms of taste. You can taste different things based on whatever. And they generated a taste map and decided that there's 5 primary tastes. Well, actually, back in his day, there was only 4. But he decided that, these taste buds are for this, and so on and so forth. I'll tell you about that in a minute. I got to tell you about the five primary taste sensations. Remember, he had primary odors. There's 7. Is that right? There's 7 odors, 5 or 7, something like that. I think 7. Yeah, was that right? 7, 5 or 7, something like that. You think I would know after teaching this for so long. But there's 5 primary taste sensations that have been identified. Some books, there's more. There's like 10 or 12 or all the way up to like, a couple of dozen. They've been arguing this for years, because detecting chemicals, chemosensory, that's pretty complex. But most textbooks, if you look at it on the average, most textbooks will say there are now 5. Actually, when I was a student, there was only 4. And they added a 5th one over here that we're going to talk about. But what are they? I'm going to tell you what they are. Now, you have these categories of taste, basically for the same reason you have the categories of smells. There's advantages for smelling certain things. Remember putrid, something that smells rotten, you want to avoid that, okay? But then at the same time, you identify certain things that have certain tastes. There's advantages for knowing that. The first one I have listed here is salty. You eat something, oh yeah, I know what something salty tastes like. See my first little picture I have right there, some Mr. Salty pretzel twists. Pretzels, everybody knows, I want something salty to snack on. Pretzels or saltine crackers or something. Salty. Everybody knows what something salty tastes like. Here I tell you, salts usually contain metal ions like sodium and potassium and so forth, which all are important electrolytes. When you get into physiology, remember we were talking about bone. I said your bones were an ion depot. Those are metal ions that are stored in your bones. Of course, you need those. And so your body can have cravings for salts. And so you'll seek out salts. Have you ever heard of a mineral lick or a salt lick? What is that? A mineral lick or a salt lick? If you have horses, or some people use them for cattle too, but we were horse people. We had horses, and we had this big blocks of salt we would put out, and the horses on occasion would go over and they would lick this big block of salt. Why do they do that? What? All animals do that. You know, they have a inert drive to acquire electrolytes in part of their diet. You know, because if you're feeding your horse, you know, the same old thing every day, it might not have all these other electrolytes in it. So you kind of like fortify their diet with that. And so that's how you do it. But I remember asking my dad, what's the real reason we give these horses these mineral licks or these salt licks? And he goes, well, we don't really need to do it, but it keeps them from eating the dirt. I said, what? He goes, yeah, because, you know, you think about it, you know, horses and other equines in nature and deer and, you know, elk and so on and so forth are herbivorous out in nature. They don't have salt licks. Where do they get their electrolytes? Well, they eat dirt. And so, you know, owning these expensive horses, you know, your horse is eating dirt all the time. It's going to acquire nasty stuff from the dirt. It's going to get worms and, you know, bad on its teeth or whatever. So it's just to keep your horse nice and fancy, right? But that's where you normally would get it. And there's actually a condition that affects certain individuals where they become, what's the term? Geophagic. Geophagia. You ever heard of that? Geophagia? You know what geophagia is? You ever heard of that? Geophagia? Geophagia, geo like earth, phagia means like to eat. So geophagia is the phenomenon in which someone has a drive to eat dirt. It's like when humans, there's certain individuals in the human population that have a craving to take a spoon and go sit out in their flower bed and start eating the dirt. Who does this? You know who does that? Anybody know? Anybody out there in Zoom land? Are you paying attention today? Pregnant women. What? Yes, there's pregnant women on occasion they crave, I need some dirt. It's a real thing. Go look it up, geophagia in pregnant women. So what's going on with that? Well, you pregnant women, you know, they have a developing individual taxing their physiology, taxing their electrolyte balance in their body. So you're going to need more electrolytes. Well, instead of going out eating dirt, go visit an OBGYN and he or she will prescribe prenatal vitamins so you don't have to go eat some dirt. Anyway, so salty, so you can detect salt. Sweet, that's a second primary taste sensation. Of course, sweetness, you know, is produced by sugars, which are included in high energy foods, you know, like a chocolate cake. Everybody knows that, right? Sour, everybody tasted something sour before. It kind of makes my mouth kind of water. Do you see me saying the word sour? Look at my little picture here. Sour is usually produced by acids, like citric acid in citrus fruits, you know, like a lemon, sucking on a lemon. Oh, man, I can just taste it now. Sour, you know, and I don't know why. You remember I was telling you that the kids have an acute taste, you know, their taste is like more powerful than adults. But why is it they like such the nastiest things? You know, they like the sour, the better, you know, the sour candies. It just disturbs me. You know, they like kids, they eat stuff like that, or they seek out bizarre types of candies, like vomit flavored jelly beans. Who would want to eat something like that? Oh yeah, give me that. Kids are weird. Number 4, bitter. Something everybody knows something that tastes bitter, right? So here I have bitter produced by alkaloids like caffeine, nicotine, and things like this, these bitter tasting chemicals. Most alkaloids exist as botanical toxins. Like the plant produces these alkaloids to make itself taste bad. So I said botanical toxins, rendering the plant unpalatable. That's the idea anyway. The plant doesn't want to taste good because it doesn't want to get eaten, right? But we don't, what do? We like coffee, you coffee beans, you get, you can take a mouthful of roasted coffee beans, it wouldn't be too good. It'd be like eating like pecan shells, something real bitter like that. Whether the idea is don't eat me, don't make a drink out of me. But we just, well, no, we're going to do it anyway. Put sugar on it, and balance it out. So anyway, but that's what it's there for. And then, like I said, when I was a student, there was only 4, but this 5th one came along, I think maybe, I don't know, like in the 90s or something. It's called umami. The word umami literally means delicious. It's the most recent taste sensation described by the Japanese, identifying a savory or a meaty flavor. And if you think about this, it makes sense. Everything that we've looked at so far is, you kind of craving this or avoiding that or whatever. But gosh, just about everybody I know that is experienced a moment when they're like wanted to eat some meat. Man, I could go for a big old steak right now or a big old fat burger or something. I need some meat, some sustenance. Let's go get some boudin, whatever, something with some sustenance to it, And so that makes sense if you want to crave that. So what's in meat, proteins, and proteins have amino acids, and you use those amino acids to build up your own proteins, right? And so here I tell you, what do I tell you? Savory, meaty flavor, so produced by free glutamates, commonly found in fermented foods like cheeses, like a lot of your cheeses complement your meats. Also mushrooms, that also complements meats. And of course, meats themselves, the sensation is intensified in the presence of sodium. That makes sense too. You add salt to your meats and you go, this really tastes good. And also MSG, monosodium glutamate. The MSG actually is, properly used in cooking to enhance the flavor of meat or make something taste meatier. That's why you can skimp on your meat, and as long as you add enough MSG, it makes you think that you're eating meat. Anyway, so there's that. Now, back to the taste buds and the different types of taste buds. And I was telling you about that guy's. It was like in 1880 or something like that. His dissertation. He developed this taste map, or the primary taste zones. And we used to teach this in phys, and we'd have a phys lab that we would map out our taste map on our tongue. You identify the primary taste zones. And here, I tell you, this doesn't exist anymore. That's why I got a big red X through it or whatever. So the taste map, or the primary taste zones, formally, not anymore, but formally showed regional differences in taste sensations across the tongue. We had sweetness in the tip, sourness along the sides, and bitterness in the back of the tongue. Where, remember, these are the circumvallate papillae back there. That's what they used to, that's what they're there for bitter, right? They had saltiness along the edges. And during the era of this taste map, umami wasn't a thing, so it wasn't included in the map. But recently, the taste map, I think probably 20 some years ago, the taste map has been discredited because all qualities of taste be elicited from any region of the tongue that contains taste buds. And as a matter of fact, it doesn't even have to be on your tongue. It can be in the roof of your mouth, your cheeks, or the back of your throat. You have taste buds there as well. And that explains why, when we would do these laboratories, and what we would have is these little litmus paper sticks, or just little strips of paper or whatever, and each one tasted like something. And so you have like this bitter piece of paper, and you're supposed to touch it to your tongue. You know, I don't taste anything. You put it in the back of your tongue. Oh yeah, I can taste it way back there. Same thing like sweet. So you put a little sweet piece of paper. You can taste it on the tip of your tongue, but if you stuck it on the back of your tongue, you're not supposed to be able to taste it. Well, we would do these experiments and they would never work. Some people thought maybe power of suggestion, they would work. But most of the time, no, I can taste sour here, taste sour there, taste sweet here, taste sweet. So it never worked. And so there finally, someone decided to investigate this guy's dissertation and look at his statistics that he was using to generate this taste map. And I guess they didn't analyze his numbers, because his numbers were all wrong, or they were not significant. And so his dissertation, he'd been long dead, but it was just BS, you know, it wasn't true. So this taste map has been existed for a long time in science, you know, but it was bogus, you know. Anyway, I forget the guy's name. I don't even know if it was in the United States. It might have been in London or something. Anyway, that's taste. Now this is our last special sense organ, but it's actually one organ, but two senses in one. So our fourth and fifth special senses, hearing and equilibrium. Of course, when you're studying these special senses, you study the anatomy of the ear. And there is your number 3. The anatomy of the ear has 3 divisions, you know. You kind of see it's kind of color coordinated. You got outer ear or external ear, same thing. You have middle ear and then inner ear or internal ear. So there's bam, 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 3 parts. And we study it like that, the 3 parts of the ear. So structures of the outer ear include, moving from the outside in, the auricle. The auricle is that thing attached to the side of your head. This is what you're looking at, this individual's auricle. Auricle. Only mammals have these structures. Then on only apes do you call it an auricle. In non-ape mammals, I guess, they're called pinnae, or singular would be a pinna. So don't point to this thing on the side of somebody's head or the side of a monkey's head or an ape's head and call it a pinna. Because the word pinna means like it's shaped like a feather, you know, in the sense that something that's pinnate is kind of tapering, kind of comes to a point. You know, like a cat has pointed ears, or a horse or a bat or something like that. They typically have these kind of tapering, pointing ears. But the ape ear is not shaped like that. It's rounded. You see like this. So that's an auricle. The word auricle comes from the Greek word that just means ear. So that's what we call this, the auricle. And then, of course, function. And why do we have these things? Well, I said only mammals have them. And of course, if you look at the hearing structure in invertebrates, it reaches its pinnacle in mammalian evolution. There's nothing more complex and effective in hearing sound wave energy than the mammalian ear. And so what it does functionally is it directs sound wave energy into the external auditory canal, your ear hole right there. And of course, it's channeling it towards your eardrum. Here I have some structures worth noting associated with the auricle. For example, the helix. The helix is this outer cartilaginous rim of the auricle. And then in from that is the antihelix, the broad inner cartilaginous rim. So you got right here, antihelix, helix, antihelix. A triangular fossa. It's like the base. You can almost see where the antihelix comes down. It kind of splits into 2. But right there is bone. You know, it's of course skin covering bone, but it's like the depression at the base of the antihelix. And then the concha. Remember conch or conch means like a snail shell. And the conch or the concha, this is kind of, we're gonna say bowl shape surrounding the antihelix. Well, it's actually the base of the helix. You follow the helix around where the helix attaches to your skull right there. So kind of surrounding right there, that's the conch, okay? And then I think this is my favorite one. And most mammologist, or it depends on what day of the week it is, whether I'm a herpetologist or a mammologist or ichthyologist or ornithologist. That's why I just say I'm a zoologist, because I do all animals. But when you're in bat mode, you're studying bats, the tragus is your favorite surface anatomical structure. So what's the tragus? What's so fun about a tragus? Well, bats, the bat ears, bats, everybody knows bats can hear really well. And the morphology of their tragus is species specific. So you can identify bat species based on the shape of their tragus. So what's our tragus? Well, our tragus isn't real important. We can't hear like bats can. So it's just this little bump right here. Tragus. It literally means goat. I guess they call it the goat. Like this is a new buzzword now in our vernacular. It's a what do you call it? What do you call a word that's created from initials? I'm missing it. It's an abbreviation for greatest of all time. Like Michael Jordan is the GOAT, greatest of all time basketball player, whatever. Anyway. GOAT. Well, this isn't for anything like that. This is, I guess, because like a goat has a goat gruff, like a beard. And I guess old men got like a hair growing from here, your bird's nest. I don't know, I'm just guessing. But this structure right here, I define it as a flappy, fleshy flap projecting posteriorly over the external auditory meatus. And some mammals, it's more functional or significant than others. Ours just kind of sits there, doesn't really do anything, doesn't move. You can close it over, if you push on it, it'll close over your ear hole. Isn't that nice? So you have a tragus, your goat, and then across from it, an antitragus. And so the tubercle across from the tragus forming the inferior base of the antihelix. So maybe here's your antihelix, you get your little bump right there. And then the lobe or lobule or just earlobe. This is the cartilage-less portion of your auricle. So there's no cartilage in here. There's it's an inferior fleshy extension of the two helices devoid of cartilage, composed of loose connective tissue. So if you open this up, there'd just be areolar connective tissue in there. Maybe a little bit of adipose, okay? So let's talk about the parts. From the auricle, you move into the external auditory canal. That's this tunnel right here, see right there? Tunnel-like chamber within the temporal bone, channel sound wave energies to the tympanic membrane. That's your eardrum, see right here? That's where it stops. And your eardrum basically is the boundary between the outer ear and the middle ear. And as far as structures are included, so where's the eardrum fall? Is it fall is in the inner ear or the middle ear or the outer ear? It's kind of like straddling the fence. Well, in my class, we're going to define the tympanic membrane or the eardrum as part of the outer ear, and you'll see why in a minute. The external auditory canal is also the location of the apical, the modified apical glands. Remember the ceruminous glands, those ones that produce earwax. So cerumen, which traps debris and repels insects. Remember we talked about all that? And then finally, your tympanic membrane or your eardrum, which is still part of the outer ear or the external ear. Why? Well, because it's like right on the middle, but we classify it as outer ear because it's an integumentary structure. Your integument's on the outside, so your eardrum is as well. So integumentary membrane closing over the middle ear cavity. So the space behind it is the middle ear cavity or the tympanic cavity, same thing. But functionally, your tympanic membrane or your eardrum vibrates in response to the sound wave energy. Okay, so the energy, it travels down here, it hits this thin membrane, and it vibrates. And we've all seen this, like a drum. How does a drum work? You strike a drum with a drumstick, and you boom, it makes a noise, right? And what's happening is, or if you watched a speaker move, it's vibrating, right? And that's, sound wave energy being emitted, okay? And of course, since it's vibrating, this initiates the stimulus of hearing. So it's vibrating and it's gonna cause other things to vibrate. So that's the outer ear or the external ear. So now structures of the middle ear. The middle ear, like I said, is also known as the tympanic cavity. The middle ear is air-filled. It's an air-filled cavity within the petrous portion of the temporal bone. So there's no fluid in here. There's not supposed to be. If there is, then there's a problem. So there's just air. Structures in it include, worth noting, or I guess probably the most important things, would be the auditory ossicles. Of course, auditory or acoustic for hearing, you know. The word ossicle means little bones, and there's 3 little bones in there. The malleus, the incus, and the stapes. The word malleus literally means hammer, like a mallet, it's like a hammer. An incus is an anvil, you know, like what a blacksmith would use a hammer or a mallet on to like making horseshoes or whatever. And then stapes, not stapes, but stapes means stirrup. And if you look closely, that thing right there looks like a little stirrup, you know, the thing you put your feet in on a saddle. Well, these little bones, they ossicles here, the ossicles conduct vibrations from the eardrum to the inner ear. So the eardrum vibrates, and the back of the eardrum is the malleus. The malleus vibrates because it's connected to the eardrum. Malleus connected to the incus connected to the stapes, and the stapes is connected to the side of your skull. And it's vibrating, and on the inside, the other side of the skull is your inner ear, where all the sensory structures are. They're going to detect those vibrations. Okay, those are the ossicles. Now, there are some muscles associated with the ossicles, and I just call them auditory muscles, and there's 2 that we identify. The auditory muscles contract in response to sounds of sustained high energy. In other words, if you're exposed to a loud noise for a long time, these muscles will contract to kind of pull the ossicles away from each other, because they're loud noise, that's a lot of energy, and that's all bam, 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 a lot of vibration. All right. And so these bones vibrating against each other, they can, I guess, get damaged. And so, and of course, it'd be overwhelming, the loud noise. And so to kind of to quail that noise a little bit, these muscles pull the bones away from each other so they can't quite vibrate against each other efficiently. And so it's less, I guess, intense. And you've probably experienced this if you ever attended a loud event, like a loud concert, or out at a club late at night with loud music, and you're partying and you're like, hey, you want another beer? What? I can't hear you. You're sitting there screaming all night long. You're having a good time. And then you leave and you're staggering across the parking lot trying to find your car. And you go, hey, you gonna drive? You okay to drive? What? I can't hear you. You can't hear it. Why can't you hear it? Well, your auditory muscles have been contracted. And so you can't hear normal sound. It doesn't, something's wrong with my ears. Oh, it's because it's all that loud noise. They've been pulled away from each other. Okay. And so over time, of course, they'll relax. You'll be able to hear normally, but they were kind of protecting your ears while you're in there partying it up. So anyway, there's 2, the tensor tympani and the stapedius. See these? The stapedius, or here, the tensor tympani, you see located right here on the back of the malleus. It inserts on the malleus, creating tension on the eardrum when it contracts, because remember, the malleus is connected to the eardrum. It kind of pulls it away. And then the stapedius right here, it shows you the tendon. It inserts on the stapes to help displace it from the inner ear during contraction. Remember, the stapes is actually connected to the side of the skull. Okay, and that's you see that like going on right there. Now, other structures of the tympanic cavity or the middle ear, the eustachian tube. This is this long tube right here, eustachian tube, or that's actually an eponym. That's actually named after somebody. The non-eponym term, I guess, is pharyngeotympanic tube. Pharyngeo for pharynx and tympanic for tympanic cavity. So it's the tube that connects the tympanic cavity to your throat. That's exactly what it is. Here I define it as the passage between the middle ear cavity and the nasopharynx specifically. It's normally flattened and closed all the time until you open your mouth and swallow or you yawn. So it opens when you swallow and closes or when you yawn. Why? What's going on with that? Well, why do you have this tube that connects to your throat? Well, it allows for drainage of the tympanic cavity. If you get fluid in there, of course, it'll drain down into your throat. But also, since it opens up on occasion, it equalizes the pressure between the tympanic cavity and the external auditory canal. Because if you think about this, out here, that's, you have air in there. And that air that's in the external auditory canal continues with the air that's out around you. And the air out around you is under atmospheric pressure. So you got so much pressure in here, but then inside here, there's a separate air space that has gas in it, air, and it's under pressure because it's separate from the outside pressure. And so if you're moved from one area of certain kind of pressure to another, your eustachian tube helps equalize that pressure. Like, for example, if you ever been in an airplane, and you get up in the airplane and your ears kind of start to hurt, and people, you can remedy that by chewing gum. What? Yeah, if your ears bother you on a plane, chew gum. What's fresh breath have to do with my ears? That has nothing to do with your ears. What do you do when you chew gum? You swallow. And every time you swallow, your eustachian tube opens up and your throat is connected to your oral cavity or to your nasal cavity, your nasal pharynx, throat, okay? It continues with the outside world. And so when this opens up, it opens up into your throat and equalizes the pressure. Because when you're going up in an airplane, it's low pressure. So the pressure out here drops. And so if the pressure out here drops, it's higher inside of here. So the pressure is increasing proportionally on the inside of your ear, and it causes your eardrum to bulge out. My eardrums are bulging. They're like being pulled out. So if you swallow, they flatten back out again. Just the opposite happens when you go diving in a swimming pool. You ever done that? Going to the deep end of the pool and it hurts your ears. And they go, you chew gum in the pool? No, that doesn't work. What do they tell you to do if your ears? They say, well, you could do that, but you better not because you're going to blow your eardrums out. Have you ever heard that? Do you know anybody who's ever held their nose and blown their eardrums out? No, there's a urban legend or old wives' tale that you do that. Like if you hold your nose and blow as you're underwater, make your ears pop, kind of remedy the sensation that you're feeling in there. What's going on? Well, when you're diving in a pool, you have water on you. And the deeper you go, the more water you have on you compressing your body. And what you do when you're underwater, you rarely have water in your external auditory canal. You usually trap a little bit of air in here. And so the air is compressed. You get water on the outside compressing that pocket of air, and that pocket of air is compressing up your eardrum. And so you get more pressure out here. It's just the opposite. You get more pressure out here than you do in here. So your eardrum is going in. And so if you hold your nose and blow, what you're doing is you're forcing air up your eustachian tube into that cavity, and you're neutralizing the pressure that way. So it's just kind of working in reverse. Anyway, that's probably more than you needed to know. So that's the middle ear. Now, structures of the inner ear. You see this blue thing, and you have it like in lab, or I'm going to show you pictures of this thing in my online lab. And I'll tell you this again, that you don't really have one of these things in your head like this. You say, what? Why are we studying this? Well, what you have in your head, actually in your, remember the petrous portion of the temporal bone? That's where your inner ear is located, okay? And the petrous portion of the temporal bone has hollow spaces and channels and little pathways that would kind of take on a shape like this. There's all the blue, okay? That's like the hollow spaces. Like if you poured plaster in there and made it like a little mold and you popped out, you'd pop out this blue structure. And so we kind of just study the structure like this and call it the inner ear, or the bony labyrinth, or the osseous labyrinth, or something like that. So here I kind of hammer all that out. So structures of the inner ear, the osseous labyrinth. Osseous means bony, labyrinth means like a maze, like a bony maze. What is the osseous labyrinth or the bony labyrinth? Or here they call it the bony labyrinth or osseous labyrinth. Same thing. I define it as complex hollow spaces within the temporal bone. Of course, it has three, right? It always has three regions. The cochlea, that's this part right here that's kind of shaped like a snail shell, because cochlea means snail. The center part right here in the middle is called the vestibule, all right? We'll use this word vestibule a lot in anatomy. Almost every body system we have has at least one vestibule of one sort or another. And what a vestibule always is like an entryway where you enter an area. And so when you enter into the inner ear, you go in through the vestibule, okay? So anyway, entryway preceding an entryway, whatever. And then the third part are all these ring-like structures. Those are the semicircular canals. And there's three: a superior, posterior, and horizontal semicircular canal. And of course, they're all connected to the vestibule. Like the cochlea is also connected to the vestibule. So the vestibule is in the middle. You got canals on one side and cochlea on the other. Now, you have openings into that. Of course, the openings are associated with the vestibule. You have an oval window, oval because it's oval shaped. It's also called the vestibular window. This is where the vibrations enter the inner ear, because the oval window is covered by the stapes. This is where the stapes attaches. And if you look at the base of the stapes, it's shaped just like that, okay? And so that's, it's vibrating. And it's vibrating right here. And then you'll see on the inside of that oval window, there's fluid that we're going to talk about. And so you got this mechanical vibrating energy. It's going to turn into wave energy again by turning these little waves of fluid inside the inner ear. There's also a round window right here underneath it. It's also called the cochlear window because it's at the base of the cochlea. It's also covered by a secondary tympanic membrane. And most books will explain that it's where the vibrations exit the inner ear. Like vibrations are coming in here, and they travel down the cochlea, and then they come out here, something like that. That's the osseous labyrinth. Now, obviously, if you looked at the osseous labyrinth, it has stuff inside of it, the actual sensory structures, because the bony labyrinth or the osseous labyrinth are just the spaces, and the spaces contain the sensory structures. And so collectively, all those sensory structures are called the membranous labyrinth. And so I define the membranous labyrinth as a collection of membranous ducts and sacs suspended within a fluid. Remember I told you about that fluid on the other side of the oval window? Well, that fluid's called perilymph. Peri means surrounding, lymph means like water. It's like surrounding water. It's inside the chambers of the osseous labyrinth all throughout. The spaces within the membranous labyrinth are filled with a second fluid called endolymph. So inside the brown, see the brown is the membranous labyrinth. All this is hollow as well. And so there's fluid inside of there called endo, like within. So endolymph is inside the membranous labyrinth. Perilymph is what's surrounding the membranous labyrinth. Does that make sense? Okay. Structures of the membranous labyrinth include inside the cochlea, you have a cochlear duct. And the cochlear duct contains the actual organ associated with the hearing called the spiral organ of Corti. And you'll study probably the function of this and how it works in physiology. I don't really get into it in lecture, but that's where it's located. It's in the cochlear duct of the cochlea. for hearing. And you know what sound is, take a physics class, they're talking about amplitude and frequency and all that kind of stuff. Well, that's interpreted by different portions of this long coiled tube. I don't get into that. But anyway, here, this is all just hearing right here, cochlea. Everything else is equilibrium. You have a saccule right here connected to the cochlear duct located in the vestibule. This round place right here is the vestibule. But this is the saccule in the vestibule, okay? It's connected to the cochlear duct. It has receptor cells located within it called maculae, which just means spots. A little green dot right there, that's a macula, all right? The maculae are for static equilibrium. You have two different types of equilibrium. There's static equilibrium and dynamic equilibrium. Static equilibrium is the equilibrium associated with motion or not. Actually, stasis means like not to move, but if something is static, it's moving, right? And so this kind of lets you know that you're in motion or not, right? And how does it work? Well, you know, the best analogy I can think of, I got almost out of time, is like one of those snow globes. You know, that's got fluid in it, you shake it up, it's got little fake little pieces of snow in it that kind of move around. That's exactly what this is. It's hollow, there's fluid in there, remember endolymph? There's also calcium carbonate crystals in there, that fake snow. And when your head moves, that fake snow brushes across the maculae and stimulates it. And that's how you know that you're in motion. That's all based on gravity. You have a saccule, and then there's a utricle. It does the same thing. It's just connected to the base of the semicircular ducts this time. But right here has a macula associated with it. It's still within the vestibule, connected to the semicircular ducts, maculae for static equilibrium. So really the whole, the utricle and saccule in the vestibule for static equilibrium. Everything else in the semicircular canals, you have semicircular ducts, but this is a different form of equilibrium. This is for dynamic equilibrium. Dynamic equilibrium is for like rotational movement, like directional. That's why they have three, because each one's in a different axis, an X, Y, and Z axis, to let you know which direction you're going. You're going up, you're going down, you're going left, right, to or fro, whatever. That's dynamic equilibrium. And the sensory structures associated with them are called the cristae ampullaries. And they're at the base of the swollen ducts, so you can hear that. Then innervation of the ear. Remember we talked about vestibulocochlear nerve, cranial nerve number 8. It's actually formed from two nerves. You have one, the cochlear branch. Well, I'll set it backwards. The vestibular branch going to the vestibule, which is for equilibrium. And then the cochlear branch or the cochlear nerve going to the cochlea for hearing. So that's why you got vestibular cochlear from the vestibule and cochlea. See how that works? The end. Now we're done with 15 seconds to spare. How about that? You guys have any questions? What about my big crowd today? Any questions out there? So we're done. And you know what? Next week, you're off for spring break. You can go test out your auditory muscles at the Loud Club. Nope, you don't do that? I don't club anymore anyway. So anyway, you can have crawfish boil or fish fry or something, do something like that. Okay, well, you guys have fun. And I will see you in week after next. Okay, no questions, I'm gonna leave. All right, bye.